Now that we have familiarized ourselves with the anatomy of the kidney and the anatomy of the nephron, let's turn our attention to how the kidney is forming the filtrate and processing that filtrate to form urine. So as we discussed earlier, the kidney is capable of filtering an impressive amount of fluid per day. We talked about the kidney's ability to filter around 200 liters of fluid per day. Now, to put that into perspective, 200 liters of fluid per day means that our entire plasma volume is being filtered more than 60 times per day. From this, we get about 1.5 liters of urine. So this is a very efficient process. So from that 200 liters of processed fluid, we're only producing about 1.5 liters of urine per day. So this is a very efficient process. It's also a very energetically expensive process. So the kidneys make up about 1% of our body weight, but they use about 25% of the daily oxygen that we take in order to make ATP to fuel this process. So it's very efficient, but energetically expensive. The three processes that we're going to be taking a look at deal with the formation of urine and adjusting blood composition. So we'll be talking about glomerular filtration, and then the processing of that filtrate that's formed by glomerular filtration. In one of two processes, the processing occurs is called tubular reabsorption. So the tubular reabsorption is the process of removing the needed substances from that filtrate. So tubular reabsorption reclaims water, electrolytes, glucose, and things like that. So we'll see that in tubular reabsorption as one part of the processing of that filtrate. The second part of the processing of the filtrate is tubular secretion, which means that the kidneys are going to be adding unneeded wastes or unwanted substances to that filtrate. So when we talk about processing the filtrate, we're collectively talking about tubular reabsorption and tubular secretion. Glomerular filtration is occurring at none other than the glomerulus, because that's where blood is being directed to the nephron in order to be filtered. Tubular reabsorption and secretion are happening within the renal tubules. So back to that original diagram that we had discussed, the structures of the nephron. Glomerular filtration is happening at none other than, guess where, the glomerulus here. The filtrate that is produced is captured by the glomerular capsule and directed towards the renal tubules here. Tubular reabsorption and secretion are happening at the renal tubules here, as their name implies. So let's take a look at glomerular filtration, right? We talked about the fact that glomerular filtration produces a cell-free and a protein-free filtrate in a healthy kidney. This is a passive process, which means that it requires no ATP because it is driven by hydrostatic pressure at the glomerular capillaries. Hydrostatic pressure is also known as blood pressure. Since the glomerular capillaries are blood vessels, the pressure inside of those vessels is what drives that filtrate into the glomerular capsule. So recall that hydrostatic pressure is the force of a fluid on the wall of a blood vessel or a pipe or something like that. So in this case, glomerular filtration is a passive process. In a healthy kidney, as I mentioned, it produces a cell and protein-free filtrate, but we still see things like glucose, sodium, potassium, electrolytes, and things like that in the filtrate. So those are all normally present in a healthy kidney. We just don't see cells or proteins in the filtrate. If we do see cells or proteins in the filtrate, then there's something going on at the nephron. There's a problem with the nephron. So returning back to the filtration membrane or the the place where filtration is occurring at the renal corpuscle. We'll look back at the junction between the glomerular capillaries and the podocytes. Right. So keep in mind here too that what I want to point out is that we've got the glomerular capillaries entering the glomerular capsule here. So the afferent arteriole is delivering blood to the glomerular capillaries. Blood makes its way through the glomerular capillaries and exits through the efferent arteriole here. And the glomerular capsule surrounds it essentially like a kind of like a hand kind of clutched around maybe a ball or something like that. So the glomerular capsule completely surrounds and encases the glomerular capillaries. And this glomerular capsule does that by the outer wall kind of folds in and on itself and wraps around the capillaries that are inside of the glomerular capsule itself. So we get this infolding of the glomerular capsule wall so that it surrounds the capillaries and collects any of the filtrate that's being produced by the capillaries and directing that filtrate into the proximal convoluted tubule there. So this is where we're going to see the filtration membrane. If we take a look at just one of those glomerular capillaries by itself, we see that the podocytes, the foot cells, surround the glomerular capillaries and extend these little foot processes out between. So you'll see these foot processes kind of link up, really kind of like loosely connected puzzle pieces between two podocytes here. And there's little gaps or little spaces in between the foot processes that are called the filtration slits here. So the filtration slits allow fluid but not cells or proteins to pass through the filtration membrane. So we did talk about the filtration membrane in a previous segment, but we'll review it here. The filtration membrane is between the blood in the glomerular capillaries and the filtrate that exists or is found in the glomerular capsule. And we see that it has three layers. It has the capillary endothelium, which is the wall of the blood vessels of the glomerulus. The basement membrane, which is a glycoprotein or sugar protein glue that binds the podocytes to the capillary endothelium. And then finally, we have the foot processes, the podocytes in the podocytes that make up the inner lining of the glomerular capsule here. So filtrate is able to pass through the pores or the fenestrations in the capillary wall, go through the basement membrane. Even though the basement membrane is a solid structure, it really does not prevent fluid from getting through it. It's really only serving to prevent things like small proteins from getting through. And the filtration slits are, again, a small little space between the foot processes, the podocytes, that allow filtrate to flow from the capillaries in the glomerulus from the capillaries in the glomerulus into the capsular space. So the filtration membrane allows the passage of really just small molecules and small substances that are dissolved within things like water. So filtration membrane allows water to pass through. In a healthy kidney, it allows glucose, small amino acids to pass through, sodium and potassium, and nitrogen-containing waste. So all of these are relatively small substances here. What it does not allow the passage of is larger cells and proteins. So blood cells in a healthy kidney are prevented from entering the filtrate. Plasma proteins are too large to pass through the filtration membrane and into the filtrate. So protein in the urine or blood cells in the urine indicate a problem at the filtration membrane. So it could be that the filtration membrane is damaged, for example, and it's allowing blood cells or plasma proteins to flow into the filtrate. So because proteins stay in the blood and do not pass through the filtration membrane, they help maintain osmotic pressure in the blood. That is, they help promote the pulling of fluid or help promote fluid being pulled back into the blood. And so we'll see that come into play later. So that helps keep water in the blood by osmosis. So there are three pressures that determine what are called net filtration pressure. And net filtration pressure is the sum of the pressures either promoting or inhibiting filtration at the glomerulus. Right? We have three pressures here, and their direction of flow is indicated by the direction of these arrows. So this arrow that's pointing to the right is indicating that this pressure is pushing fluid out of the capillaries and into the glomerular capsule. These two arrows that are pointed to the left here are indicating pressures that are pushing or pulling fluid into the glomerular capsules into the glomerular capillaries, I should say, from the glomerular capsule. So these three pressures are as follows. HPGC is the hydrostatic pressure of the glomerular capillaries. So that's the blood pressure that's in those glomerular capillaries. Osmotic pressure of the glomerular capsule is OPGC. And HPCS is the hydrostatic pressure of the capsular space. So let's take a look at what all three of those pressures are. So let's start with hydrostatic pressure in the glomerular capillaries, or HPGC. If you want to think of this in sort of simpler terms, this is really just blood pressure, right? So blood pressure is the main driving force that's pushing water and solutes like glucose and sodium and potassium into the glomerular capsule, right? So this is the blood pressure of the glomerular capillaries that's forcing fluid out of the capillaries and into the capsule. Hydrostatic pressure of the glomerular capsule is about 55 millimeters of mercury in a healthy kidney. So recall that our units of pressure were in this measurement, or in this unit called millimeters of mercury here. And this is maintained in a healthy kidney at about 55 millimeters of mercury. Now, I want to make a note here that this is higher than in most capillary beds. Most capillary beds exhibit blood pressures or hydrostatic pressures of about 26 millimeters of mercury. So this is a little bit more than double what you would see in a normal capillary bed. And this is due to the fact that the efferent arteriole here, so the blood that's leaving the glomerular capillaries, is smaller in diameter than the afferent arteriole. So you've got a lot of blood volume coming in through the afferent arteriole. Nice large diameter afferent arteriole is promoting a lot of blood flow into the glomerular capillaries, but we have a small blood vessel draining it, or the blood vessel that's leaving the glomerular capillaries is smaller in diameter than the blood vessel that's entering it. And so this promotes a high blood pressure in the glomerular capillaries. So that is the hydrostatic pressure in the glomerular capillaries pushes fluid out of the blood vessels and into the glomerular capsule. We'll see that these two forces, the next two forces, are pulling water back into the glomerular capillaries. The first of those forces is the hydrostatic pressure in the capsular space. So this is the pressure, the fluid pressure of the filtrate that's occupying the capsule. So this pressure forces water and filtrate back into the glomerular capillaries. The second of these pressures is the osmotic pressure in the glomerular capillaries. And this is what is known as the pull that's produced by osmosis. So recall that in a healthy kidney, proteins remain in the glomerular capillaries. Large proteins do not enter the filtrate. And those proteins that stay in the blood act as kind of an osmotic force. So the high concentration of protein that's in the blood helps to osmotically pull water from the capsular space back into the glomerular capillaries. And so we see this osmotic pull. And so for that reason, the osmotic pressure and the hydrostatic pressure of the glomerular capillary are pressures that are promoting fluid staying in the glomerular capillaries. So this hydrostatic pressure of the glomerular capillaries forces fluid out at a pressure of 55 millimeters of mercury. Osmotic pressure inside the glomerular capillaries pulls fluid back in at 30 millimeters of mercury of pressure, and the fluid that's already in the capsule exerts a pressure, an inward pressure or a negative pressure of 15 millimeters of mercury into the glomerular capillaries here. So net filtration pressure is the outward pressure minus the inward pressures here, which means the outward pressure is our 55 millimeters of mercury here. It's our hydrostatic pressure of the glomerular capillaries. And that's subtracted from the hydrostatic pressure in the capsular space, which is our 15 millimeters of mercury. And we also subtract from that the inward osmotic pull of fluid from the capsular space, the inward osmotic pull back into the glomerular capillaries there, and that's 30 millimeters of mercury right here. So the way they have this equation set up is just fine, but you can also calculate this equation by taking the outward pressure and subtracting both inward pressures. So this would, if you took 55 minus 15 minus 30, you would still end up with a net filtration pressure of plus 10 millimeters of mercury. Now this plus 10 millimeters of mercury means that overall there's 10 millimeters of mercury of pressure that's forcing fluid out of the glomerular capillaries and into the capsular space. If this was negative 10 millimeters of mercury, it would mean that this pressure is driving fluid from the capsular space back into the capillaries, which that would inhibit the ability of the kidney to do its function. So this has to be a positive outward pressure, again, in a healthy kidney, should be somewhere around 10 millimeters of mercury.